Chemistry really is at the heart of biochemistry, and biology for that matter, because these amazing processes that we're studying are the result of chemical interactions. And they're the same types of chemical interactions that you learn about in chemistry. They're involving the same atoms, the same subatomic particles. It's just that in our cells, they're often being done with the help of teeny tiny organic chemists called enzymes. And so typically in biochemistry, we're relying on watching those them do their work and trying to figure out how they're doing their work. Whereas in chemistry, you're typically focused on making things do work in specific ways. So we have some less control in biochemistry, but it's really important that we know what is going on at the molecular level and at the chemical level, and really try to understand how these chemists are working. And so having a solid understanding of chemistry is really going to help you. Even though they're involving the same types of reactions, and sometimes they start with the same things and end with the same things, enzymes often have the reactions happen in a slightly different way, and are able to really speed up reactions. And enzymes can even make possible reactions that organic chemists can't do. So we can learn a lot about chemistry from biochemistry, as well as chemistry being important for biochemistry. So if you want to major in biochemistry or even biology, you typically have to take chemistry. Like, we had to take it before we took any biology classes. And you might be wondering, like, why the heck am I doing this? But do not just treat it as a prereq that you've got to pass. It's seriously, if you understand these concepts, they're going to show up everywhere. Chemistry really doesn't need to be scary. It can be fun, and understanding it in an intuitive way can help you connect concepts and build upon the foundation of general chemistry through organic chemistry into biochemistry, into biology, into all of these different areas. And so I want to help try to show you how you can develop this intuition and how that will help even these really complex things seem simpler and more understandable. Chemistry can keep sounding scary, especially organic chemistry, but really I've been thinking a lot about it recently, and you can really condense it down into some core principles that if you think about things in that way, you can see that these principles will go from the level of electrons, so subatomic particles, all the way to like whole people. And so I want to introduce you to these concepts, as well as show you examples of how they come into play at the different levels, as well as how chemistry's been really useful and how these concepts have been really useful in my own research. And point you towards some resources that can help. Okay, so these aren't like official core concepts or anything. They're just things that I found helpful. And I'm just going to try to give you a sense of the things that you should be keeping in mind, or that you might want to keep in mind. I'm not like telling you what to do or anything, but things that might be helpful to keep in mind. And then we'll look at some instances where they come into play. Okay, so the first is that everything wants to move as much as possible. So things really like to be free, and they want to move. And we're talking everything by thing. Like, I'm talking down to the level of subatomic particles, so electrons, all the way to atoms, to molecules, to heck, whole people, right? I mean, like, I like to move. I'm sure you like to move too. Talking about moving, like, this can mean like be referring to, say, the movement of electrons to form new bonds. So I'm not just talking about like molecules just moving around, which is part of it too. But other things make this difficult for us, and for these things. So one of them, the main one we're going to get into more in detail, is the opposite charges attract, and like charges repel. So talking about like electrostatics. So if something is getting, it's trying to escape, but it's getting attracted by another molecule, so it's sticking to that thing, or maybe it wants to go this way, but there's a negative charge, there's a same charge thing there, so it's getting pushed away. So basically the movement can be hindered by this electrostatic. It can also be hindered by steric hindrance. So basically things can't occupy the same place at the same time. So if two molecules, if you have a molecule like this, and then they can't like get close enough to actually like interact, or they can't move in the way that they want to move. And so no matter what the like hindrance, it takes energy to move. And so the bigger,
the harder it is to move, the more energy it's going to take to do so. And the more energy it takes, the less likely it is to happen, and the more stable it is currently. And this energy can come, so in like when we're doing chemistry and stuff, this energy often comes by like heating up a reaction or that sort of thing. But we can't really do that in, our cells can't really do that. So instead, they do things like they can get energy by pairing reactions to the release of energy from ATP or something. And so basically we have like high energy things are less stable. So the concept of energy can get really confusing. But when we're talking about like energy, we're typically talking about like potential energy. So it's easiest to think about potential energy in forms of like potential kinetic energy, where you have like a bowling ball at the top of a hill, and then it drops down. And so up here, you would say this is high energy because it has high potential energy. And this would be low energy. And so it's releasing this energy as it goes down, and that energy can be used to do other things, like powering reactions. So we see this a lot in biochemistry. But it's the same concept. So like basically when things have the energy to do things, they'll do things. Because remember, things like to do things, they like to move, they like to do things. And so when I talk about move, I'm not just talking about like movement, I'm talking about things like also movement of electrons. So like movement of electrons to form bonds, and to, so that movement actually can involve interactions and reactions, not just like a molecule floating around. But basically, so when they do things, when they can do things, they will do things. And there's a lot of ways to do things, right? So if you have energy, if you have money, there's a lot of ways to spend that money. And so you're typically going to spend that money. And then, so things tend to be in the no money state, and it's harder to get money, right? So when things are more like stable, it tends to be in this like low energy state. And when they're in this state, now they don't have energy, they need to do anything. So it's kind of like if you were to look at the end of the day, most people would have spent their money if they could. They might have spent it in different ways, but they'll be in this more stable state. But sometimes to get to like the optimal stable state, so the optimal low energy state, reactions need help. And so there's often like a transition state, like a high energy transition state, and you need an activation energy to get past that barrier. And so in chemistry, and in biochemistry, this can be so in addition to like we talked about before, how you could like add energy, so such as like adding ATP, using ATP, energy from ATP, hydrolysis in biochemistry, or in chemistry using like adding heat or maybe pressure. But we can't do those things in biochemistry, remember. And even in chemistry, you still might need to speed things up in a different way. We'll do this with catalysts, which are things that speed up a reaction without being used up in the process. And so in biochemistry, the catalysts are enzymes. And so these are typically proteins, sometimes a combo of proteins and RNA. And what they do is they provide like an alternate path from the high energy state to the low energy state, or it doesn't actually have to be like this. You can be going from, there's all sorts of different reaction energy curves or whatever. But the point is that at the end of the day, you're going from the same amount of energy to the same amount of energy, whether or not the reaction is catalyzed. But when you're catalyzing it, you're kind of providing a different way. Or somehow stabilizing the transition state, doing other things to provide an easier route that takes less energy and makes things more likely to occur. So this is the key point, is that just because something is like wants to would be more stable in this other form doesn't mean they can make it there. And just because it would really, really rather be here doesn't mean it can get there, because it needs to get over that transition state. And so that's why enzymes are super, super helpful about doing these things. So just because something can happen doesn't mean it will happen. And often you need like a catalyst or something to make things happen. And in biochemistry, reactions are often coupled so that you can use the energy from one reaction to power another reaction. Okay, so some more core concepts. Opposite charges attract. And so this is what's going to keep the protons, so these positively charged protons in the nucleus are going to rein in these negatively charged electrons. This is why sodium and chloride form a salt. This is why glutamate and lysine, so these amino acids, so these parts of proteins, can form these like salt bridges that like can provide part of structure. Then like charges repel. So if you have like same charged amino acids, then they'll repel. And so this can help change the dynamic. So different proteins have different shapes depending on these things. And if, so say an amino acid gets phosphorylated or whatever, now you're adding negative charges. And so you can modify the things with these charges. And it's really crucial is that this is true of partial charges as well. So as we'll see, molecules can share, can have these partial charges. And when they have these partial charges, this allows them to do partial charge interactions. And these interactions can even be like temporary and induced and random. So that's another key point is that things are probably probabilities and things are probably random. And so when we're talking about numbers and like whether or not a molecule will be this way or this way, we're usually talking in terms of probabilities. And so, and like, so even when we're talking about like binding constants or whatever, we're talking about like the concentration at which half of it will be at something or whatever. And it's not like you could just know like which one is which or whatever. If you were to, it's not like you could say select one of them that you knew would be one way. It's saying like this molecule has a 50-50 probability of being in another way kind of thing. So just keep that in mind that things are probably probabilities. Okay, so going back to the key concept of things wanting to move. And so this is often coming down, we can think about this in the terms of electrons. This is also bringing in that concept of the opposite charges attracting. So atoms are made up of protons, neutrons, and electrons. And so you talk about these a lot in general chemistry. And in biochemistry, we typically focus on the electrons, which are these negatively charged particles. And you can think of them kind of as really energetic dogs. And they're getting reined in by the protons, which are kind of like the dog walkers. And so the protons are these positively charged parts. They're kind of getting glued together by these non-charged neutrons. But so that's forming this dense central nucleus. And then the electrons are whizzing around in a cloud. And so here's where, for another instance of probabilities, is that we can define like where these electrons will probably be, like more where they're most likely to hang out, but we can't actually predict where they'll actually be. But what we can say is that, so these, they want to, they're like trying to pull away from the protons because things like to be free, remember? So it's not like they don't like the protons, they just want to be free. And so they're going to be like pulling away, and the protons are going to pull them in. And the farther away they are, the more energy is required because they're having to pull tighter on that leash. And so we have the higher energy electrons are going to be further away. And if we can provide even more energy, sometimes we can even pull them off. And if they find like a better option, they can actually like pair up, and this is how you get bonds. So you can think of like the dog walkers coming together to share the responsibilities, but they don't always share fairly. And so like if you have in water, the oxygen is more electronegative, so it's going to pull better on those electrons. And so even it's going to kind of take some of the hydrogen share. And so you have this cloud of electrons, and now the electrons, the probability of finding an electron is going to be more likely near to the oxygens. And this is going to leave you with polarity. So you have this partial charge where the oxygens are partly negative and the hydrogens are partly positive. And now we have, again, we have our opportunity for charges for partial charge attraction. And so not that one. So water can form these intense networks, and they can also, since they have those positive and they negatively charged regions, they're really good at dissolving other things as well. So they can form these networks with themselves, but then also with other things. So water is really good at dissolving things. So another place we see that these partial charge attractions is with like hydrogen bonding. And so really this is just a partial charge attraction, because the oxygens are going to be partly negative. So basically you just need something like, so these are going to be partly negative and these will be partly positive, and then you get these interactions. And so this is what keeps our DNA held together, and it's also a key part of protein structure. So like these beta plated sheets and these beta strands and these alpha helixes, these are held together with hydrogen bonding. And so this, remember, is a partial charge attraction. It's just a more stable one than like a stronger one than ones from, say, like induced dipoles. So basically, because we have those electrons, like remember, we can't say like where exactly they are, but we can give like a probability of where they're likely to be, but they're like whizzing around. And so sometimes they're moving around like randomly. And so they're sometimes going to be more likely to be in one area than another. And if, say, a charged thing comes by, that can shift their electron density, because then the electrons that were near that charged thing will either be repelled or attracted based on that whole like charges repel, opposite charges attract thing. And so this can cause like a temporary shift in and make even non-polar molecules, so molecules that were sharing fairly, now they're partially charged. And then you can even get these networks. This can even happen instant, like just by chance, and then you get these like chain reactions. And this can allow even like geckos to walk on walls and stuff. So it's pretty amazing. And all of this is just these partial charge attractions. And then at the like strongest end, you have ionic bonding, which is like full charge attraction. All right, going back to that steric hindrance thing. So this comes into play a lot with protein structure. And so proteins are made up of these long chains of amino acids, and then they fold up to give you these pretty proteins. And there are 20 different common amino acids, and they have these different side chains, so these unique R groups that stick off. And you can see that some of these are really bulky, and especially like these branch ones that have this branch really close to their to the backbone, they're going to provide they're going to make it harder to move. So whereas ones like glycine, which have just the hydrogen, they have a lot more flexibility. And so this is just we call a Ramachandran plot showing kind of the angles of the backbone. So if you have something like a valine, which has that branching, the chain, it can't move as much, or else those groups are going to like bang into the backbone. And so you can't have that. You also, if you have like, so basically molecules need their space. And so even though when we show like crystal structures of proteins, so we show those like pretty models, if you show like the space filling model, it's really not just like a stick, it's like a more of a blobby thing. Not a blobby, but I mean, it takes up more space than you would expect if you just look at that. Okay, what else did I want to mention? Speaking of those amino acids, so a lot of these, if you're taking chemistry, these groups will seem familiar, and especially organic chemistry. So we have all of these different like functional groups, which are kind of like, so organic chemistry, basically you have like this carbon hydrogen backbone, and then you can have these functional groups which is where stuff happens. And so the same functional groups that you learn about in organic chemistry and that you may manipulate in the lab are the same functional groups that are crucial to the reactions occurring in our body. And so you can see that like kinases, so a type of enzymes, they're using like these reactions. They're making these mechanisms. You have this arrow pushing all of this stuff that you would learn about in organic chemistry. Is happening inside of cells and the test tube when you have like enzyme in there or whatever, it's happening. So that the chemistry is coming into play everywhere. So also going back to things liking to be free. So we talked about the electrons wanting to be free. And so we can also see that we can excite electrons, so such as light absorption. So if electrons have more energy, they can get farther away. They can like pull their leash further. But the energy electrons, they have to be occupy like distinct energy levels because they have these like wave-like properties, and so then they're like quantized. So they can only occupy distinct energy levels. But if you have the right amount of energy to get them to what their next energy level, then you can absorb that energy, and you can get that energy from light. And so electrons can, if they get higher energy, absorb light. But then remember, what you absorb, you must then release. And so sometimes this is released in the form of light, and then we call that like fluorescence. And so we can take advantage of these processes and make cool reactions and label things and that sort of thing. Other things that like to be free. So we can talk about at the molecule level. And so we have things that if you have enough energy to escape those interactions with other things, then you can like fully become a gas. If you don't have as much energy, you can only slide around, then you're a liquid. And if you really don't have enough energy to move, then you're solid. And so these this is like the energy, and then so you can think about water's great escape. So it's trying to make it out and all of these attractions around it. Remember, because the positive, the partial positive, partial negative parts, then also has this pressure pushing down. So it's hard to move, but it's trying to be free, remember. Sorry, today's post is a little all over the place. I just wanted to give you a sense of some of the ways that chemistry comes into play in biochemistry, and how sometimes it's similar to what you might have learned in chemistry, but it's also can be slightly different. We're typically dealing with like only a subset of the elements. And so they're typically like similar, most the same as what you learn in organic chemistry, because organic chemistry is basically doing very similar reactions often to the type of reactions that our bodies do, but they're doing it in different ways, like without the help of enzymes and stuff, but using like the same molecules sometimes even. And so these primarily just have these elements. So you're only going to have to worry about this stuff most of the time, although it helps to know about especially some of the things with metals, because often, for example, enzymes might use metals as well. So here's a practical example from what I was doing today. So I was doing some slot blot experiments, which is a way that you can measure binding affinities. And so this, you have a nitrocellulose membrane on top and then this nylon membrane on the bottom. And then so this is used for like protein, RNA, or DNA binding. And so you put the nitrocellulose is on top and then the nylon's on the bottom. And when you apply a vacuum, the liquid's going to go through, and any protein and any bound RNA or DNA is going to get stuck on top, and then the rest will flow through to the bottom. And this nylon, which is positively charged, is then going to trap the negatively charged nucleic acid. And so there you have your charge attractions. And so to actually get this, I radio labeled the nucleic acid. So I put like So you do see like radioisotopes. So focusing on like in general chemistry, you learn about like nuclear isotopes, and so like having different numbers of neutrons. So that's where that sort of thing comes into play. Also the thermodynamics you learn about. And so for these experiments, what I'm doing is I'm measuring the binding affinity, which depends on how the difference in, since I'm doing it at equilibrium, it's going to depend on the difference in like stability or the difference in energy between the bound and the unbound forms, and like which is preferred. And so this is going to depend on those like reaction equilibriums, constants, and that you learn about. You can also look at kinetics, so how fast the reaction is, if you want to look at like the on rate and the off rate. So all of this is going back to things you learn about chemistry, especially if you're talking about like enzyme kinetics and you're looking at like Michaelis-Menten kinetics. So all of this stuff that you learn in chemistry really does come into play. So it's really important to have a good understanding of all of it. So yep, this was me this morning, current mood, but hopefully this video will help someone just like see kind of how chemistry comes into play all over the place, and kind of the core things to keep in mind when you're going through, and to try to focus on some key concepts. So this post was actually inspired by, I found this really great article, Essential Chemistry for Biochemists, which is very, very helpful and geared towards biochemistry. And I highly recommend that people read it. It's good review if you are in, if you're like doing biochemistry already. It's also can help you, if you're starting biochemistry, know what types of things you're going to encounter. I also really like ChemGuide UK. It's this really great website from Jim Clark, and some really, really good in-depth things about chemistry concepts. And then Khan Academy has some really great videos. So I hope that something here will help. And now I need to get back to analyzing this, the binding data.